Okay, now that you have this carbon cycle handout ready to go, let's take a couple of minutes and label what you need to label on this diagram. It's the same as the last diagram on your notes. This one's just a little larger, gives us a little more room to make a little more, detail information. So we're going to start right in the middle, Calvin cycle, for our label of what this is a diagram of. And then we will go to the top where our reactant is entering the Calvin cycle. And again, this is our carbon dioxide reactant, and there should be 3 of these. They enter one at a time. As they enter, they meet up with this molecule called rubisco. They are then combined and rearranged with rubisco to form this, what they call short-lived intermediate. It's not around for a long time. And this occurs in phase 1, carbon fixation. So it is taking those carbon molecules from the carbon dioxide, fixing them, rearranging them, putting them into a larger molecule than what they started out. This is the steps it takes to make our glucose ultimately. So we now have a 1, 2, 3, 4, 5, 6 carbon molecule, and there are 3 of those. And that is rearranged into 6 3-carbon molecules. Here comes our ATP again, our energy from the light reaction. Broken down, use the energy to rearrange these carbons to make these much larger carbon molecules. And that continues throughout the carbon fixation cycle and into phase 2 of this Calvin cycle. And during phase 2, we get our introduction of NADPH. Phase 2 is called the reduction phase. And it's doing exactly what it sounds. It's reducing. It is taking these large carbon molecules and reducing them down to our ultimately G3P molecule here to get them to be the size that they need to be to ultimately end up being our glucose molecule. Don't forget though, NADPH is going to be used as part of that process. Some of the hydrogens are going to be used to help make the G3P, glyceraldehyde 3-phosphate. Remember that 6 of those are made. One of them, right here, one of them will leave. This is that G3P or P-gal molecule here that's leaving. That goes through another series of changes to ultimately become glucose. We don't get into the details of how that becomes glucose, but it is ready at this point to be made into a glucose molecule. The other, and the rest of those G3P molecules, the other 5, move on to phase 3, which is regeneration, or also rearrangement. I hope I spell that right. Rearrangement. And again, it's now taking these 3-carbon G3P molecules, rearranging them with the help of energy from ATP, more of the ATP that was made in the light reaction, using that to make those now 1, 2, 3, 4, 5 carbon molecules that combine with the CO2 to start this process. This 5-carbon molecule is our RubyP, and that is short for ribulose biphosphate. Ribulose biphosphate is RubyP. RubyP becomes rubisco. Rubisco combines with CO2 that enters to once again start the Calvin cycle, producing the glucose molecules that we get from photosynthesis. Okay, so now let's take it to one last paper. That one last paper is this giant overview of photosynthesis that we started last week, the week before. And we want to just add some details about the Calvin cycle. We've filled most of this in already, and I'll give you a minute to find it. And if you need to, at this point, you might pause the video so that they can find what they need. So we filled in on this the equation, the light-dependent reaction. We did a simple modified Z diagram. We talked about the water that comes in and the oxygen that comes out in this first portion of photosynthesis, the light reaction. We looked at how that produces the ATP and the NADPH molecules, which then are used by this light independent reaction, dark reaction, or Calvin cycle. We had already filled in the fact that the CO2 enters the light independent and that glucose leaves the light independent reaction, and that this takes place in the stroma. What we want to do is just one more time make a simplified version of what is happening. Say simplified, it might get a little complicated, but we have our cycle, so we have our circle, and we just kind of want to divide it into 3 parts, something like this, where step 1, step 2, step 3. In this case, I'm not being precise, and the size doesn't mean that one is that much bigger than the other or that much longer. It's just the way I drew it. So remember, carbon fixation is phase 1. And phase 2 is reduction. Excuse me. And phase 3 is regeneration. And so here's where maybe some of the confusion is going to come in terms of craziness of this diagram. But remember, our CO2 is going to enter right here in the carbon fixation stage. You're going to have ATP coming in kind of at that reduction / carbon fixation stage, and being broken down into ADP plus PI. You're also going to have, at this point, the NADPH coming into the reduction area, or reduction phase, and it kind of kicks out that NADP plus. Ultimately, here we go, by the end here we get 6 G3P or P-gal molecules by the end of the reduction phase. One of them leaves the cycle entirely as the 1 G3P or P-gal, and then that ultimately leaves and makes our glucose molecule. The other 5 G3P molecules will continue on to regeneration. More ATP comes in to the regeneration phase to be used for energy, which makes or leaves ADP plus a free-floating phosphate as the end result. And here, those 5G3P will meet up with Ruby P. Right, so we kind of draw an arrow, it's going this direction, meets up with Ruby P up here, and that will continue on into the carbon fixation cycle or phase when carbon dioxide is present to continue making glucose as long as you have CO2. And that's it. Those are the notes, handouts, diagrams that we need to finish photosynthesis. I have a couple of review handouts that we'll go over, but I'll probably wait until I get back for that. You are now going to work on some practice pages. You're going to do some handouts that need to be turned into your folders. You need to make sure that you do them by yourselves. Not talking, not sharing answers. You've got a ton of notes in front of you. Use them to help you complete these activities and handouts. They should not be difficult, they should not be hard, but they should help prepare you for your photosynthesis quiz that you have on Wednesday. Even if I'm not here today, you still have that quiz on Wednesday. Make sure you study your notes. You just simply need to know what we've covered over photosynthesis. It won't be too detailed, mostly kind of what goes into each part of the chloroplast, where it occurs, what leaves, your equation, basic definitions. It will not be multiple choice. It will be short answer. It will be you filling in the blanks. It will be you having to know photosynthesis. So you have to do something called studying and taking these notes home. Do not leave them in your green folder. The only thing that should be left in your green folder will be the notes or the practice pages that you turn in to be graded. They're not important that you take them home to study from because you've got plenty of other things to use to study. I will be here on Tuesday. I will have advisory and before and after school tutorials on Tuesday. I will also be here Wednesday morning. If you have questions, need help, or just ready to study and want me around while you're studying for your quiz. But you do need to make sure that you're studying and you are prepared. Be good. Do not give this up a hard time. I will see you guys on Wednesday.